So good afternoon, everyone. My name is Stephen Firth. Thank you for joining us for the Monash Advanced Imaging Series. Today we have Rob Parton with his talk, Cave Exploration at the Nanoscale, Multimodal Molecular Imaging to Study Membrane Microdomains. Rob studied biochemistry in the UK before moving to EMBL in Heidelberg. He received Royal Society and EMBO postdoctoral fellowships before becoming a group leader in 1990, studying plasma membrane domains and cell surface dynamics. In 1996, he moved to the University of Queensland and is currently a group leader and division head in the Institute for Molecular Bioscience and deputy director of the Centre for Microscopy and Microanalysis. Today, he's going to talk to us about the use of light and electron microscopic techniques, including electron tomography, serial blockface scanning electron microscopy, and genetically encoded electron microscopic tags to gain a quantitative understanding of the formation of specific plasma membrane domains and their role in endocytosis, mechanoprotection, and specific signal transduction pathways. So to our audience, please put any questions in the Q&A or chat window, and we'll attend to them at the end of the seminar. And thank you very much for agreeing to speak with us today. And over to you, Rob.
Thank you very much, Stephen. So I will, I'll turn off my video and I will share my screen and then we'll get underway. Okay, everybody hear me okay? So what I'd like to do today is to talk about our research in the lab and highlight some of the techniques that we're using along the way. And our aim is really to understand various aspects of cell biology. We have a number of different projects in the lab. We've been recently working on lipid droplets as antibacterial agents. We've also taken 3D data of a migrating breast cancer cell. We've converted that into a VR experience. And I mention these because if anybody wants to ask me about these, then please feel free to at the end of the talk. But what I'm really going to concentrate on today, as Stephen said, is how we are studying plasma membrane microdomains. And this includes various different microdomains. It includes microdomains at the nanoscale, such as RAS signaling nanodomains, at the mesoscale, caveolae and clathrin-coated pits, and also at a much larger scale, we're also interested in the T-tubules that penetrate into the depths of muscle fibers. But really today, I'm going to focus on caveolae, the formation and the function of caveolae. I'm going to describe what they are, and then I'm going to talk about how we've studied them. And then I'm going to, at the end, for the second half of the talk, I'm going to mention some of the methods that we've developed or more correctly adapted for high throughput 3D localization by electron microscopy and how that correlates also with light microscopy. And what we try and do in the lab is to really go from molecules, including atomic structures, to organelles, to cells, and then into animal models as well. So the methods we use have to be useful across these multiple scales. So these are caveolae. This is a beautiful image sent to me by John Heuser, and you're looking from inside a cell at the inside of the plasma membrane, and you can see these pits. These are invaginations of the cell surface. You're looking at these little caves from the inside of the cell. And so these are caveolae, and you can see how abundant they are. So they're about 60 nanometers in diameter. And they can be incredibly abundant, as in the last slide, or they can be fairly sparse, as shown in a standard cultured cell. This is just a BHK cell in culture, where caveolae can occupy a few percent of the surface. Caveolae can also be present as these single structures, as shown here, or they can be in these complex arrangements. These are all caveolae in these kind of rosettes. Now, caveolae density really varies across different tissues. I'm going to show a couple of examples. This is muscle, where you have a capillary, and you can see in the capillary wall here, the caveolae, which I've highlighted in red. And one function that's been proposed for a long time now is that things are transported via caveolae across the wall of the capillary. This is another example of a cell type where there's abundant caveolae. This is white fat from the mouse. These are two cells. This is the huge lipid droplet in each cell. And then you have this thin layer of cytosol. And then attached to the plasma membrane, you have these caveolae, as you can see here. The other tissue that we're particularly interested in, and we'll come back to this a couple of times in the talk, is skeletal muscle. And there are various muscle diseases associated with dysfunction of caveolae. We study adult muscle fibers, where you have this very complex arrangement of filaments, but also the membrane systems. And we can label the surface with electron-dense dye. And when you do this, you can then use electron tomography to look at the organization of the surface. And this is a tomogram. It's a 300 nanometer section. I'm just going to run through it, and I'll point out a couple of things. But what we're starting from, we're sort of outside the cell. We've labeled the surface with electron-dense marker. As we go through, we can now see all these caveolae in this complex arrangement. As we go deeper, we can now see these T-tubules. These are tubules that go down into the depth of the muscle. We're coming back out again, and we're thresholding these structures. And now again, you can see the caveolae, and you can see how complex these structures are. So the caveolae really increase the surface area of the muscle. This is a completely controlled wild-type muscle, and they make this very specialized organization driven by the caveolae. So very abundant in certain cell types, linked to transport in some cells, endocytosis, signaling, mechanosensation, and mechanoprotection. So a specialized form of signaling where you're sensing mechanical changes at the cell surface. So I'm going to introduce the major components of caveolae. These are the caveolins and the cavins. And I'll talk about how we believe, using various techniques, as I'll gloss over as I go through, how they generate a metastable surface domain. So the caveolae looks like this very rigid structure, but we believe it's actually a regulatable domain that can be disassembled in response to certain stimuli. And then I'll talk about the functions of caveolae, or one particular function of caveolae, and that's in mechanoprotection, and a new story which hasn't been published, where we have a new model for modulating cell shape and looking at the role of caveolae. And then I'll finish with these methods for high throughput 3D localization. So starting with the caveolae then, the caveolin is a small integral membrane protein, about 20 kilodaltons, about 150 of these in a caveolae, so very abundant, and linked to various diseases such as lipodystrophies, where you lose adipose tissue, and muscle diseases. And we believe that caveolin is embedded in the membrane somehow, with the N and the C terminal on the cytoplasmic side. But until recently, we had no real structure of the caveolin. And there are 3 caveolins in mammalian cells, caveolin 1 and 2 together in non-muscle cells, and caveolin 3 in muscle. And we know that if we knock out caveolin 1 or caveolin 3, we lose caveolae. But we also know that caveolin itself can drive formation of invaginations. And I'm going to show a couple of examples. This is all published, so I'll go through it quickly. First of all, we showed in a cell-free system. So this is a leishmania extract. We can add DNA in this mixture. It makes RNA, it makes protein. And then the caveolin is actually inserted into the membrane. And I hope you can see here, this is actually tomography of a negatively stained sample. So even though this is negatively stained and a very thin sample, the tomography allowed us to look at high resolution. And you can even see this GFP caveolin. You can see it's actually concentrated where these buds are. So this technique is actually, this is embedded in a methyl cellulose layer, is actually allowing you to see a GFP tagged very small protein without any other label, although we have a couple of gold particles there. We can also express caveolin in another system. And again, this is quite intriguing. You can take E. coli, no internal membranes. You can express in this system, express the caveolin. And what happens is the caveolin inserts in the membrane, the cytoplasmic membrane shown here. This is a fast frozen free substituted cell. And it then oligomerizes and generates vesicles that we call heterologous caveolae. So these caveolae accumulate inside the cell, and we use those for various things. We can actually purify them. We can use them as a nanovesicle for drug delivery in cells and in mice, and we also use them for vaccine development as well. So these vesicles, this is a purified prep, negative stained specimen. You can see how uniform they are. And recently, Anne Kenworthy and Melanie Owie managed to use this system. They deterged, extracted these, and got the cryo-EM structure of caveolin. So it's a beautiful bit of work, not our work at all. We're very pleased that they used our system. But they got this very nice structure of caveolin. It looks quite similar in this cross section to what I showed earlier. But in fact, if you look in 3D, what happens is that the caveolin is forming an oligomer. This is an 11mer, and it's sitting in the membrane. The N termini are here, the C termini in the middle. And so this remarkable structure is what's driving the formation of caveolae in these model systems. So it's an incredible structure, and we're still trying to understand how it all works. Now, even though the caveolin disc, as I've shown here, can drive caveolar formation in these model systems, in a vertebrate cell, you also need another set of proteins. And I have to introduce those now. These are the cavins. So these are peripheral membrane proteins, not integral. We think they have an elongated structure. They are very much concentrated on the bulb of the caveolae. This is immunogold labeling shown here and here. And this is also labeling on a plasma membrane lawn. They're using 2 nanometer gold. And at this very high resolution, you can see these kind of striations. And you can also see this in deep ETM. So we think the cavins are actually forming these striations on the surface of the caveolae, on the cytoplasmic surface of the caveolae. So a little bit about the cavins. The cavins are very charged. They have these helical regions, which are very basic. And then they have these disordered regions, which are very acidic. There are 3 cavins in muscle and non-muscle cells, so ubiquitously expressed, whereas one of them, the cavin 4, is specific for muscle. And these proteins form a coat complex in the cytosol. It's then recruited to the plasma membrane, and that recruitment process requires the caveolin, caveolin 1, caveolin 3, and cavin 1. And again, various diseases are associated with mutations or loss of the cavin proteins. So we've solved the structure of part of the cavins, particularly cavin 1 and cavin 4, and in this helical region, which mediates oligomerization of the cavins. That's homo-oligomerization and hetero. It's a trimeric coil. It has a pi45p2 binding patch, and this binding patch is a sensor for membrane association. When the protein comes off the membrane, it gets ubiquitinated in this region here, and then it gets degraded. So it makes sure that there's very low levels free in the cytosol. The protein cavin 1 also has a unique domain. We think this is vital for forming caveolae. And that's an 11 amino acid repeat domain. So it's an undecad repeat. And we could show that that domain binds phosphatidylserine. So the cavin 1 has these 2 distinct lipid binding sites. And so this is what we think. This is just a model. Only part of this has been solved. But we think the protein through these lipid binding domains is sitting via the helical regions on the membrane. And then these disordered regions are looping out into the cytosol to interact with the caveolin and to mediate oligomerization. And so I won't go through this. This is recently published, beautiful work by Vikas Tillu. And this is all a collaboration with Brett Collins' lab. So we don't quite now know how it all works. We have the discs, we have the filamentous cavin. These come together. But with Nick Ariotti now, we have very nice data by cryo-EM of very thin cells grown on grids and then fast frozen, where we're now getting very nice cryo-EM data on these caveolae in situ. So we want to be able to now dock these kind of structures into the cryo-EM structure in cells in situ to understand how this system works. So this slide summarizes the main points. The caveolin gets made in the ER, passes through the Golgi, and then at the surface, you get caveolar formation. And this relies on specific lipids, which we've characterized in this paper here. The caveolae can also be internalized and recycled back to the surface. But what we think is a crucial part of the function of the caveolae is that they can also be flattened. And this can happen in response to specific mechanical tension on the membrane. And then the other part of this is that we think that when they do flatten, the cavin proteins can be released into the cell. So these are the key concepts. This was quite a bit complicated. Multiple low affinity interactions between the caveolin, the cavin, and membrane lipids. Then you make this metastable structure. And then in response to physical tension on the membrane, this thing can disassemble and you release these proteins into the cell. And I'm going to show a couple of experiments on this mechanical aspects here. So what we speculate in cells with abundant caveolae, that if the caveolae flatten out, that will prevent the membrane damage in response to this increase in membrane tension. So it's like a reservoir of membrane, like a little spring in the membrane. And so we looked at this in a couple of systems, and I'm going to very briefly mention this story where we looked at the zebrafish notochord. So why is the zebrafish notochord, and what is the zebrafish notochord? So the zebrafish embryo is able to swim around because it has, at this stage, it has no kind of skeleton, but it has a very rigid structure in the middle. This is a tube which is under hydrostatic pressure, and then that pushes out against the sheath that surrounds it. And this is the notochord. So it's a rigid tube in the center of the zebrafish embryo. And this is a beautiful structure for electron microscopy and it's absolutely full of caveolae. So this shows a tomogram here from fast frozen cells, which have been free substituted, but the caveolae are incredibly abundant on this membrane, which is within the notochord. So we thought this was intriguing. It's really a mechanical structure, and it's one of the most abundant sources of caveolae that we've ever seen. So what we did was to knock out the notochord cavin 1 in the zebrafish. There are actually two, but at this stage, there's only one expressed in the notochord. So we knocked out the cavin 1b, as it's called, and we could show that this caused a dramatic reduction in caveolae. And this is shown here. This is the wild type. This is the knockout. Not only were there less caveolae, but the caveolae that remained had this very distinct shape. Instead of having this very characteristic flask shape, they were now much more shallow, changed in morphology. So what would this mean for the function of the notochord? What we saw in the knockouts was that over time, we got these lesions in the notochord. And these are areas of damage. And we could actually watch this by light microscopy and see that these cells were actually bursting in real time. And this accumulated over time, as can be shown here. This is just two different measures of lesion formation, damage to the notochord. And we could also show that there was increased damage if the embryos were moving around more. If we immobilized the zebrafish embryos, there was very little damage at all. So it really was a case that the lack of intact caveolae, or the reduction in total caveolae, was causing a problem with the ability of the notochord to actually withstand these pressures. This could be rescued by the wild type cavin 1, but actually not by a mutant construct which lacked this repeat region that binds phosphatidylserine. So in the zebrafish notochord cavin 1, different from human, there are actually multiple of these repeats. And we could show, and this is work from Vickers, shown here, that these repeats actually made the caveolae more stable. So we could do cellular experiments, and we could show that if you had more of these repeats, the caveolae were more stable. So mammals, such as Vickers, shown here, 2 repeats, zebrafish has 5, and in evolution, there seems to have been a need for even more repeats. The mud skipper, for example, has got 6 of these repeats, which we would speculate would make even more stable caveolae. So the caveolar flattening maintains the membrane tension, we think. It allows the cell to change shape, and that means that in a cell with abundant caveolae can occupy up to 50% of the cell surface. If they flatten out, that can allow cells to change shape. And this is just another example of this. You can take fibers from mice, these are adult mouse fibers, put them in a hypertonic medium, very severe condition. The wild type fibers get some blebs, but they don't burst. But the fibers that lack cavin 1, when you put them in this hypertonic medium, not only do they contract, but they actually burst open, the calcium rushes in, and then they contract down. So our model is really summarized on this side. On this slide, we suggest that the loss of caveolar proteins changes the membrane fragility. So you have no caveolae. When there's these shape changes, the caveolae would flatten out. But this is only a model. There could be many, that's our model, but it really is just our speculation. Because is there any way we can actually see whether this is true? Whether under cases of extreme shape change, that the caveolae would be flattened out. And so what we wanted to find was a system, preferably an in vivo system, where we could get a defined cellular shape change and then look at what happened to the caveolae in that system. And so this is unpublished work, but it was really stimulated by the fact that when we're looking at our zebrafish or other fish, you can see that they go through these incredible changes in body shape. And when you have a curved fish like this, then one side of the fish is compressed and the other side is extended. We could also show this with our zebrafish embryos. We did very high-speed photography to look at this, and you can see this incredible curvature of the embryos. And what we thought was, could we actually find a way of fixing the fish in that curved state, then look at both sides of the fish and see if the muscle was different on the two sides and whether the caveolae would actually be different in the stretched side versus the compressed side. So can we trap the fish in a curved state? And we can do that because the fish normally, they're in their eggshell, they will burst out around the 72-hour stage. But if we bleach the chorion, the eggshell, so we can just put the eggshell in bleach for 5 minutes, it makes that eggshell very tough. And so then the fish can't actually escape, but they're still alive. They are flicking from side to side. So it's a trapping, but it's an active trapping because the fish are still able to move. But if you look at them at any one time, you can see that they're either at one side, as shown in green here, or at the other side. So they're flicking from one stage to the other. So that's what we did. We bleached, and then we left the fish in there, and then we fixed, and then we looked at the muscle. And what we could see, you can measure the length of the sarcomere. And so that's the length of the muscle fibers, basically, the contractile unit in the muscle fibers. So very nicely, what we saw was on the stretch side, the sarcomere length has actually increased. On the other side, the compressed side, it's not contracted, it's same as the control, but now it's very different from the other side. So you have this significant change in the length of the muscle fibers on the stretch side versus the contracted side. And I should say that the fish are still healthy. You can release them from the egg after 3 days and they will still swim away. Okay, so that's what we did. We took our fish, we bleached the chorion at this stage. Then we went for another 24 hours. And then what we did was we wanted to look at the muscle in the same fish. And so we used serial blockface scanning electron microscopy to section across from one side of the fish to the other. So we went through the stretch muscle to the notochord in the middle here, and then through to the other side. So we could directly compare in the same fish the number of caveolae. This is one of these kind of experiments, serial blockface scanning electron microscopy. And Nick Ariotti managed to optimize techniques, although they're still laborious, where he could model, he could count, basically, he could map all the caveolae in these muscle fibers. And this was a monumental effort. It was semi-automated, shall we say. But in one of these myotubes at this stage of development, 3 days post-fertilization, there's about 20,000 caveolae per myotube. So it's a massive number of caveolae. Okay, so that's what we did. That's the test to show that we can model the caveolae. And I should say that they were shown in yellow here. So these are all the caveolae that you can see in yellow that are mapped across the surface of the muscle. But then we turn to the real experiment, and this is one of the examples here. So we're going to start the periderm here. We're going to section through, collecting an image every 100 nanometers as we section the material. So the serial blockface SEM, so it's actually using a knife to section through. And what I'll do is I'll just scan through this movie, and you can see the curved fibers deeper down and the straight fibers near the top. So here we go. So this is the stretch side. You can see these very stretched muscle fibers. We're now going further down, and we're going to get into the notochord in the middle of the embryo. So this is now the notochord that you'll see here. And then we'll go through the notochord, through to the other side. And then you'll see the muscle now appearing on the compressed side. And I hope you can see this curvature of the muscle, because this is now doubled up and bent over on the compressed side. So Nick did his magic, and he did this on multiple embryos, supreme effort. And what we saw very nicely was that on the stretch side, consistently, we have less caveolae in the same region as we do on the compressed side. So these are three completely independent fish, but lots of different regions within each fish. And we had to optimize conditions so we could look at large areas and yet be able to clearly define caveolae. And we know we are defining caveolae because we have a knockout of caveolae, and we can see that we lose caveolae in that knockout using the same conditions. So caveolae is defined by morphology, reduced on the stretch side of the zebrafish embryo. And then we could compare this to the shape changes in the muscle. So we have the stretch side, the compressed side, and then Tom Hall was able to then reconstitute by light microscopy using area scan. He could look at the stretch side, the compressed side, and look at the changes in the surface area of the fibers. And we're getting this is still a little bit preliminary, but it's looking like this is going to be close, that we have about a 2.5% of the membrane surface area would be in the flattened caveolae, and that's corresponding very nicely to this apparent surface area change in the fibers. So this flattening of the caveolae is allowing you to get these shape changes. And we know this is important because if we have our knockouts, this is the cav minus here, then under these conditions where we're trapping the embryos within the chorion, then you get more damage in the knockouts as compared to the wild type. So this is combined data here, so you might want to look at this. In the bleached chorion fish, you get a little bit of damage in the wild type, but you have far more damage when you have your knockout where you have no caveolae. So this shows that not only are the caveolae flattening out, but this mechanism is important in terms of making sure that the membrane isn't damaged. And in the long term, this is a nice system to understand all the different mutants of caveolin 3 that are associated with human patients. So we think this is what's happening, that under extreme conditions, the caveolae can flatten out. We'd like to be able to look at this in real time in the fish, so we're working on that now. And we think that these superstructures that I showed in these first slides are actually important in the mature muscle fiber, because these rosettes can move into the membrane in a very efficient large-scale manner to change the apparent surface area. So caveolae required for maintenance of the notochord cell and muscle fiber integrity during mechanical stress. The caveolar flattening occurs in cells undergoing these extreme shape changes, and this obviously has an evolutionary advantage during larval stage, for example, during swimming, and in fish undergoing extreme activity. And we're now looking at this, if this also plays a role on a more a less extreme scale in terms of muscle hypertrophy in response to exercise. So I now want to move on to this idea that the cavins are signaling molecules. And so our model is that the caveolae can flatten out. This can protect the cell, but it also releases the cavins into the cell. And this was really a major goal because of observations like this, where cavin 4, this is the muscle isoform of cavin. It's on the surface of the muscle fiber. But if we look at muscle fibers lacking cavin 1, where you have no caveolae anymore, and you don't have the anchoring of the cavin 4, now you see that the cavin 4 isn't on the surface, but it's in the nuclei. So is this reflecting something that would happen in a cell when caveolae disassemble in response to a physiological force? How is it getting there? What's it interacting with? And so that's the idea, that the cavins would interact with these targets inside the cell when the caveolae are disassembled. So to do this, we carried out an proteomic analysis using biotin ligase, using nanotrap, and particularly we expressed cavins in cells that lacked caveolae because we wanted to see what these potential partners would be inside the cell, not just at the surface, but inside the cell when they're released from caveolae. And we could then identify a range of potential interactors using these methods. We could use cell-free expression and look at interaction between the cabins and target proteins, and we could quantitate that, and we could validate that by FCS, single molecular fluorescence spectroscopy, and we could then look in cells. And then once we had these targets, we could ask what happens in cells. Are there certain cellular conditions where the caveolae is disassembled? We think it's not just mechanical. We think there's other stresses that cause the caveolae to disassemble, and what do they interact with? So, and to do these, we wanted to really start to localize things in the cell. And we needed to do this not on a one-by-one basis, but really in a high throughput manner. And I've done a lot of immunogold labeling over the years, proteins like RAB5 and RAB11, we localized those for the first time. But this is a laborious process, and we wanted to see if we could use other techniques. They should be quantitative, they should be high throughput, and we should also be amenable to using them in animals and also in 3D. And so we've been using Apex, and this is not developed by us. This is a beautiful paper by Alice Ting's lab, where they showed that this small peroxidase that they called Apex could be tagged on, fused to a protein of interest, and then produce a DAB electron dense reaction product. And so this is a very nice, easy way of tagging a protein with something that you can detect by EM. And we've tried various small tags. For us, this is the only one that really works consistently. So you can tag your protein directly with Apex. We find that sometimes perturbing for localization. And we're also interested in finding a system that was compatible with our existing cell lines and animals that had GFP tagged proteins. So could we have a system for EM localization where we actually had a system where we could detect GFP tagged proteins of interest? And so this is the idea. You'd have your GFP tagged protein of interest. Apex would be recruited specifically to that GFP tagged protein. So you wouldn't need to make a new fish line. You wouldn't need to make cell lines. You wouldn't need to make new constructs. So we use nanobody. We use a nanobody against GFP, and we know a lot about nanobodies. This was characterized structurally by Brett Collins, our collaborator. And what we did was to attach the nanobody to the apex, and then we co-expressed with our GFP tagged protein of interest. And this worked surprisingly well. We find beautiful localization of multiple proteins. In a few days, we could do 13 localizations. We took a whole family of lipid binding proteins, and we could find by EM and by correlative light microscopy where those proteins were localized by the presence of this electron dense reaction product. We also developed an mCherry nanobody as well, and these are all on Adgene if people need them, which also worked very nicely and also could be used for this same kind of approach, and that's published a couple of years ago. So we have nanobodies against GFP, we have nanobodies against mCherry. The system worked very nicely in cells when we just co-transfected things. Could we also use it in animal models as well? And this is work of Tom Hall. And so he developed a couple of systems. He developed zebrafish lines where he had the apex, we use apex 2, fused to the nanobody under one ubiquitous promoter or a heat shock promoter so you can switch it on with temperature. And in the fish, there was also a reporter which is mCherry under a crystalline promoter. That should actually say Chris for the crystalline promoter. So this is the experiment. We have our zebrafish, our existing zebrafish, which have got the GFP tagged protein of interest. We cross those with our line, which have got the red lens reporter, this crystalline reporter, and they have the apex nanobody expressed as well. And then in a few days after they've bred, we have the eggs, we have the embryos, and the fish that have the GFP and the red eye are those that we're interested in, because they're expressing both the GFP tagged protein of interest and the nanobody attached to apex. And this, from the individual fish lines, once you've got these, you can cross this line with any of your GFP tagged fish lines, and you can get localization by EM in vivo in a week in the early embryos. So this works very nicely. This is just an example. This is a CAX reporter. This is a control. And then this is the apex nanobody when we've crossed the fish, and you can see this recruitment to the plasma membrane of the muscle and also to the T-tubules as well. We could also do this with the heat shock inducible lines. So we can do heat shock, switch on expression in a few hours. So you're not getting this expressed all the time. You switch it on specifically, and then you can get your localization in your tissues in vivo in a few days. We're also, though, interested in, so this depends on the nanobody being recruited efficiently to the protein of interest. So you need good expression. We wondered if we could adapt this further for low abundance antigens. And so for this, and I should have put the reference on here, but it was a very nice eLife paper where they developed a conditionally stabilized nanobody against GFP. And so what we did, this conditionally stabilized apex GFP binding protein, it's targeted for degradation in the absence of GFP or YFP. And so it gets degraded. But if you do have expression of a GFP tagged protein, it's stabilized, and then you get this specific recruitment of the nanobody. So it's stabilized by binding the GFP. So that means you get rid of this background if you don't have any unbound apex nanobody in your cells or in your fish. And again, this works very nicely. This is the apex GBP alone in these cells. You just get a little bit of degradation that you can see some reaction product. If you express with soluble GFP, with surface GFP, or with endosomal GFP or caveolar GFP, you now get stabilization, and then you're seeing apex specifically in the right place in the cell. So it's not being degraded anymore, and this reduces the background dramatically. You can also use this for other purposes, because this GFP nanobody only will bind a recombined split GFP. So the split GFP system relies on close proximity of protein X and Y. That brings these two fragments of GFP together, and then you make this functional fluorescent unit. But also, when it's generated, it now is binding the nanobody. So this enables us to actually localize protein complexes, not just proteins. And this is just one example, proof of principle, where we tagged these two parts of GFP, fragment 1 and fragment 2, to two caveins that we know come together to get this nice reaction product specifically on the caveolae. You can also use this system for other purposes. I won't go into this in detail. This is all published, so please have a look, because this, again, works very nicely. You can use this conditionally stable nanobody to also bring a biotin ligase in proximity to the GFP tagged protein of interest. And we've published this. We continue to use this method because it works surprisingly well. And so this gives you a means of doing proteomics on your GFP tagged fish lines, again, in a very time efficient manner. And again, happy to talk about this if people are interested. So Apex is a simple genetic tag for 3D-EM. It's high resolution. We can use it for endogenous level detection. We've used genome edited cells. It's compatible with high throughput EM, and we've got both GFP or M-Cherry systems. We've used conditionally stable nanobodies, and it's compatible with split GFP. And I want to finish with this final story from a great research assistant in the lab, James Ray, where we decided that what we wanted to do was to go further with the Apex and convert the diffuse, perhaps you might say non-quantitative signal you get with Apex into a particle. So can we convert the Apex reaction into a gold particle, for example? And that's what we've been able to do. And I should say we really spent a long time making sure this was a very robust method. So if anybody tries it and has problems with it, please let us know, because we think this is very robust. And it's a very simple method, really, because although there was a lot of work involved in getting it really reproducible, what it involves is doing the DAB reaction, and then we can do a silver enhancement where the silver ions are reduced by the DAB polymer to produce silver particles, and then we do a gold toning. And this is a very old technique where you replace the silver with gold. It's from 19th century photography, so we couldn't find any references for it except for these very, very old methods at the start of photography. And there's a few other tricks. We use gum arabic and other means to make sure that we have a very uniform nucleation. And you can see that the particles produced are very clear. The signal noise is great. We can do thresholding. We can use automated analysis because it's good enough for these techniques with machine learning. The other thing that was really exciting is a great collaboration with Peter Gunning and with Edna Hardman. And what we did was we convinced Peter and Edna to make a knock-in mouse with apex, the tropomyosin 3 gene being replaced by an apex tagged form of the tropomyosin. And this is actually expressed not at native levels, but below native levels. So we got the fibroblasts from these mice. They're absolutely fine. They're running around. And you could see by light microscopy after the DAB and then the enhancement, the gold enhancement, that you could see this signal. This is the electron microscopy now, that's light microscope. And tropomyosin, this actin binding protein, actually labels, we could see individual fibers that are labeled. And we did a lot of quantitation on this system and various other systems. And what we now know is that if you have the antigens well spaced out, then you can actually get to single molecule detection. So it's very, very sensitive. Of course, we chose a protein that's expressed at relatively high levels, but this is actually below the native levels for the tropomyosin. So we have very good sensitivity and we have very good detection. So we're very excited by this. We're using it in a lot of different studies now in the lab, and we hope other people can use it as well. So in summary then, I hope I've given you some examples of the imaging that we're doing in the lab, a lot of tomography, a lot of 3DM, serial blockface electron microscopy, and the applications to the formation and function of caveolae, and with a view, like many people, to moving now to cryo-EM in cells so that we can correlate structures from various techniques with what we're seeing inside the cell. And then I also described these methods for high throughput 3D localization by electron microscopy. We believe it has high sensitivity, it's efficient, and even given a single molecule resolution. And I'll finish there. We've got fantastic collaborators. Brett Collins, Vickers did his PhD with me. He's now working with us, with Brett and Nick Ariotti. Nick did his PhD with me, went to UNSW, back to UQ, and it's fantastic to have him working with us again. But he has his own group and he has his own projects, but it's just great to have him along the corridor. Tom Hall, James Ray, couldn't do anything without these guys. And also Harriet with all the skeletal muscle work, Ween in the zebrafish, and many other people here, Nick Martell for all the cloning, which is essential for what we do. People past and present, some of these you'll know. Rachel did a fantastic job working with us, and Nicole Schieber, who was at EMBL and came back to UQ. But lots of people involved, lots of great collaborators, very grateful to the CMM, Microscopy Australia, and funding bodies. And again, if anybody wants any of these constructs, please give them a go. All available to researchers through AGI. And I will finish there, and I will stop sharing, and I will start my video. Thank you very much.
Wow. Thank you, Rob. That's amazing. The specificity, that labeling at the end there with that apex is just beautiful.
It's working really well, actually.
I'll start off with a question while we're waiting for people to put some in the chat. That trapped fish in your mechanoprotection part there, you said the fish will swim away. Is there anything wrong with the fish after that? I mean, that looks like some fairly severe, for me, not a zebrafish person, it looks, some fairly severe problems with that fish.
Yeah, so what we can do is, so I think, I'd say that there is some loss of fish when we go for a long time after the bleaching. If we were to bleach and leave them overnight, we can break them open and they can swim away. I think they'd have no problems at all. The longer you go, the more immobility there is, and they're still growing, of course. But even so, you can still release them after a couple of further days, and yeah, maybe they won't develop to the same extent because they've had this whole developmental period where they've been in the chorion, but they survive okay. Yeah. Okay,
but that loss of the caveolae is that's not damaging over long term.
So so what they would be doing, that's a good question, because what's happening actually is that they're constantly flicking from one way to the other. So even though at the time we fix, we have one side where they have the caveolae flattening, when you watch them in real time, and we've done quite a lot of this, they will stay in that position for a little while, and then they'll flick across the other side. So they'll be relaxing and sort of compressing the different sides all the time. Because I think you're right. If they went long enough in that state, then there would be.
It's a bit of flipping side to side.
Yeah,
Important
yeah.
point. All right,
Yeah.
we have a question from Peter. I'll allow you to speak. Go for it, Pete.
Oh, wonderful. Hey, g'day, Rob.
Hey, Peter, great to see you, Pete.
Wonderful to see all that stuff. My head is racing, particularly with the EM silver gold replacement. It seemed like alchemy to me, but
Yes.
I wondered, you mentioned you have several different types of nanobodies. Then I began to wonder whether you could get EM resolution on the individual particle sizes or different enzymatically recognized controlled systems for making different particles. Is that possible?
Yeah, it is possible. There are some methods that Mark Ellisman pushed forward with a different system, which might be compatible with this, so that we could do double labeling. What we can certainly do is we can have gold particles internalized, and then we think we can then do the silver gold, but instead of gold, use cerium. And then we can detect that in the microscope. So we can distinguish gold that's been internalized from what we're doing in terms of the enhancement. The double labeling, I think, in the same cells at the moment is going to be challenging. And we think we could do it, but what we don't think we can do at the moment is do it in a way that is really amenable to standard cell biology experiments. You can find ways to do it. But so what we're doing at the moment is we generally have one marker plus something that's internalized or something like that. And we're also doing a lot of CLEM as well. So we'd have a fluorescent marker and we'd have the apex gold, and then we'd be able to correlate like that. So that's the way we're getting around it. But ideally, we would have two different nanobodies and different enzymes. Not quite there yet, Pete, to be honest.
And the Apex Gold protocol, is it published somewhere?
Yes, it is. Yeah, okay. So it's on in the eLife paper, but if anybody wants to try it, we've had people from all around the world contact us. Of course, probably the ones that got it to work don't contact us, but the ones who've had a few problems, and we've been able to test things and sort things out.
Brilliant. Thanks a lot, Rob.
Okay, we have another question from Thomas. Are you okay to ask this question, Thomas?
Oh, if the internet works. Great talk.
There we go. We can hear you.
Okay.
Thank you.
Just on the nanobody apex, is it possible to detect proteins within organelles? Does it get across membranes?
Yeah. Alice Ting has done that. We've certainly used nuclear localization signals to get things into the nucleus. We've not looked at anything in the mitochondria, for example. We have used apex on the other side of the membrane to tag the extracellular domain of something that was internalized. So I think it could be done, but we've not done anything with it. But I think Alice Ting did do some mitochondrial ones.
Cool. Thanks.
Okay, more questions from the audience. Anybody else, please stick your hand up or put a question into the chat for us. Chat is disabled. Sorry, a question into the Q&A. That's the way to go. All right, well, I think if we're quieter there, we can say thank you very much to you, Rob, for a fantastic talk.
Thank you, Stephen. It's my pleasure.
Hopefully that has stimulated a lot of people. That red lens reporter seems like a fantastic thing. Not that I'm particularly part of the zebrafish community here. Maybe it's well known, but that looks fantastic. So thank you very much for being with us this afternoon, and have a great evening.
Thank you very much. Thanks, everybody. Bye.
Cheers.